Good afternoon, everyone. Hope that you guys are having a good good Wednesday here. I've got good news for you guys, and that good news is that I was able to resolve the issue that I was having with my tablet. So that means that I can do what I wanted to do in terms of instruction. Um, I've got today's PowerPoint posted and the PDF. I've got tomorrow's PowerPoint and PDF posted, so those are up. I'm expecting to have Fridays before, I would say before tomorrow's class period. So you guys can kind of get a little bit ahead there. Um, I'm in the process of writing the quiz that's on Monday. I'll let you guys know before the weekend. Maybe here's some areas to study for that quiz. Emphasizing this, emphasizing that, as opposed to just saying like, well, it covers everything that we've covered so far. Um, with that in mind, though, I would say today's instruction a little bit more than yesterday's. Today's going to get a little bit more into um, what we're really interested in as biochemists, because one of the things that we're very interested in as biochemists is what a protein looks like. And that ultimately relates to the ionization state of amino acids. We're not getting into amino acids today, that's going to be tomorrow, but we are going to be touching on ionization of different acids and bases and talking about acids and bases. It's a little bit of a revision, not revision, but a little bit of a review back from Gen Chem 2, but this is a very important review refresher sort of thing. So, um, I'm going to go ahead and get cracking. If anyone has anything that they would like to ask or add, please don't hesitate to add it to the chat. In the event that you ask something in the chat and I don't respond, I've got a couple of thumbnail videos open for folks. Turn your camera on and go like this, or you can obviously unmute yourself. Okay, so I went ahead and I left my red scribbling there as proof that, hey, good news, my tablet's working. Because that's one of the things that I like to do in this class is I always like to like mark up my PowerPoints. So today we're going to be wrapping up intro stuff and then we're going to be getting into a discussion of water and ionization and water and what an aqueous solution looks like and how it impacts different aspects of life. So, to wrap up the things from yesterday, what I wanted to talk about were a couple of different things. Um, I wanted to bring up the extremely important molecule DNA or deoxyribonucleic acid. And this is probably something that you've heard of before if you've taken a like organismal or a functional biology class. But this is a very important experiment at the heart of biochemistry. This is commonly known as the Hershey-Chase experiment. And the Hershey-Chase experiment was conducted by this guy right here, Alfred Hershey, and this woman right here, Martha Chase. And what they were looking to do was basically prove once and for all. It had been kind of speculated, but they wanted to prove once and for all that DNA, in fact, is the carrier of genetic information and that it was not, in fact, proteins. So I guess they were just looking to kind of like close the door on this conversation. And in order to do this, what they did was they used bacteriophage or bacteriophages. I'm going to say phages, even though when I was in grad school, I remember there were always people who were like, phage is plural. You can't say phages. And I was like, well, you guys are much more authorities than I am because they worked in a phage lab. And I'm not looking to have any sort of fight, whether it's like a physical altercation or just like verbal. I'm not looking to have a fight over phage versus phages. At any rate, bacteriophages are effectively, you could kind of think about them as viruses that infect bacteria. And this is a good model of, or a nice crude model of a bacteriophage. And inside it has DNA, or it has its genetic material that it transfers on. And so what Hershey and Chase did was they used radio labeled proteins to make these bacteriophages. And they labeled two different things. One of those things was sulfur, because sulfur is not found in a DNA molecule. They knew that if they could transmit, if they could infect these bacteria and they carried this radio label, so basically, if the cell down here, the infected cell, infected bacteria, and I gotta say, I might need to fine tune my writing because it's usually not that blurry. And I can't help but imagine that someone's thinking, wow, he was really worked up about ensuring that he would get his tablet so that he could write really blurry letters. I try to not write such in such a condensed way. But we had infected. Let me do it this way. Infected bacteria. And I'm going to just leave bacteria down there. So we had infected bacteria, and what they were looking for was, well, if in fact, contrary to what people believed at the time, if in fact protein was the carrier of genetic information, and these bacteria down here that I put the little star in had that radio label, well, then they'd be able to say, oh, well, the radio label went with the infected or went with the infected bacteria. But instead, that was not the case. And instead, what they did was they did centrifugation to separate the remnants of the bacteriophage, circled here, separated that from the bacteria. And they found that, well, the radio label did not go with the bacteria at all. So the bacteria were indeed infected by the bacteriophage, but no no radio. Okay. Now, that was basically a binary check for, well, is it protein that carries this on? Then they did another aspect or another angle of this, and they used radio labeled DNA with a radioactive phosphorus isotope. Well, that radioactive phosphorus isotope, they let the bacteriophage incubate with the bacteria. The bacteriophage infected the bacterial cells, and then they did that centrifugation step and they separated. I'm just going to do a nice centrifuge tube here. And I would imagine that, yeah, the bacterial cells, I'm going to say bacteria, ERIA. The bacteria is there. And then at the bottom would be the phage. I'm going to say phage remnants. And I presume that's how it would separate because the phage is probably going to be super dense and it's going to go to the bottom of it's going to make up the pellet. So they looked for it and they said, oh, well, the phage remnants at this step. Well, the phage remnants, not radioactive. Meanwhile, the bacteria, well, the bacteria was indeed radioactive. And if you're thinking about this, wondering like, wow, that's pretty cool. This was also the inspiration for the Imagine Dragons song Radioactive. Not sure if that's true, but people say that. People say that. So, most importantly, though, the Hershey Chase experiment kind of solidified and confirmed the notion that DNA is the carrier of genetic information. So DNA will transmit information from generation to generation. And it was Hershey and Chase who were responsible for that. Now, we'll talk more and more about DNA and the structure of DNA, and we'll get into all of that. But I want to highlight some of the basic information, in part because we'll need it as we proceed through the class, but also this is just good foundational information for you kind of moving forward as a biochemist. So I think most folks know just through like pop culture, maybe seeing Jurassic Park or a movie, that DNA forms a double helix. Now, that is typically the way in which it's formed. And, if you've taken a biology class at some point, maybe you contrasted that with, well, RNA doesn't form a double helix. RNA is single-stranded. And, that's a nice approach. That's a good general rule of thumb, but that's not as concrete as you want it to be. It's not that simple, but that's just kind of the nature of any sort of biology. As soon as you think you know something, then it's like, oh, not so fast. But DNA forms a double helix. And one thing that's kind of like a detail or an anecdote that people feel comfortable with, but they don't necessarily know the why, is that DNA has an overall negative charge. Now, the why is because it has what's known as a sugar phosphate backbone. And the key that I want you to look at with that is when you see negative, I want you to think, oh, phosphate. Does anyone remember um, phosphoric acid? Phosphoric acid is H3PO4. Phosphate, a phosphate ion, is PO4 3 minus. So it's, again, we'll get deeper into this later on, but anytime you hear that, oh, it's got an overall negative charge, well, that's because of phosphate, a phosphate ion that helps comprise the backbone of a DNA molecule, a double-stranded piece of DNA. Now, okay, so our, kind of background basic information, we know that DNA is the carrier of genetic information. Hershey and Chase kind of confirmed that. It's got DNA is a negatively charged molecule, and its structure is a double helix. We'll get into the nuts and bolts of that. I think that it's on exam number 2. But for the time being, let's go ahead and proceed from here. Now, the last important and significant discovery related to DNA that I want you guys to know about is the Michelson-Stahl experiments. So this fellow on the left-hand side is his name is Matthew Michelson. His name is Matthew Michelson, and this is Franklin Stahl. They are actually both still alive. I always think that it's neat whenever I've heard about an experiment for a long time. Like, oh, I heard about this when I was a sophomore in high school. And it seems like it was, you might as well tell me that, oh, whenever they were opening up King Tut's tomb, that's when Michelson and Stahl were working together. And I'd be like, oh yeah, sounds good. And then whenever I learn that, oh no, that happened in kind of recent history, I'm always like, my perception of time is distorted. At any rate, these two fellows are still alive. I believe that the background there is and this is just a little bit of an aside, but Cold Spring Harbor Labs, if you ever have any interest in research related to genetics and genomics or DNA and DNA damage, CSHL, Cold Spring Harbor Labs. That's a research facility that's on Long Island, which growing up in Central Texas, my concept of New York was, oh yeah, New York City. They got the Yankees and the Knicks. And oh, there's Long Island. Oh, it's an island. It must be very small. Well, Cold Spring Harbor Labs are on the very far east side of the island, and basically the backdrop of it looks like this. It's not quite so grainy. I think that I think they have enough pixels that if you go there, you'd be able to just see leaves and everything. But it's kind of like, oh yeah, it's in New York. Okay. Upstate New York? Nope, the other part. Oh. Oh, so New York City. Well, not quite. And yes, I am revealing that I am not a Geography master. Okay, so Mieselson and Stahl, what they wanted to learn about and what they sought to determine was the way in which DNA is replicated. And they and the collective at the time had basically three models for replication. One model for replication, I always just like to think about kind of a progression. One model was termed conservative replication. And what that meant was basically you had your two strands of DNA, and you would use each of them as a template for new strands. So we could think about it as strand A and strand B. You'd separate those two strands, copy A, copy B, and then take the two copies and put those together, and then return original strands A and B to their original partners. Now, I like this because anytime that I've talked about this model or these models, it's like, well, how could it be anything but what it actually is? Because that model is like, wait a minute, you're going to break a bunch of bonds, a bunch of hydrogen bonds, separating these two polymers, and then you're going to make two new polymers, and then you're going to break them again just so you can reconstruct. Well, that's a little bit odd. But that's the conservative model of replication. Another model for replication was that basically a piece of DNA, a piece of double-stranded DNA will get replicated. So we'd have this, and then you'd have that sort of conservative model where you'd again separate these two pieces and you'd kind of pepper it all together. So you'd have something that looks like, I'm gonna write O for the more robustly read parts for the old or the original strands, and then N for our new strands. And so in your kind of daughter generation or your next generation of DNA double helices, you'd have something that's part old, part new. It's kind of like at a wedding, something old, something new, something blue. I don't think they had anything blue though. So there's dispersive, there's conservative, and there's dispersive. And both of those models include kind of using strands as a template and then making new ones and then breaking and then reassembling. So it's almost like you could think about it as break, make, break, make. Okay. Now, ultimately, the model that is the accepted model for DNA replication is what's known as the semi-conservative model. And it's a little bit tough to tell because you've got these strands right here. So I'm going to put an X and then I'm going to switch colors because I want you to kind of track this strand. It's a double helix, and a DNA double helix, it's going to twist and contort itself around a central axis. Boom, boom. So it kind of looks like a really fun roller coaster because, yeah, it goes down and then it goes up, but there's also like a twist in there. So essentially, the semi-conservative model for DNA replication was you have two strands, and both of those strands act as templates for a newly produced strand. So this would be, well, break, make. So you'd have all of these enzymes, these biological things that would come along and they'd see this single strand of DNA and they'd complement. Now, if some of this terminology in kind of visualizing this, you're like, oh, I'm having trouble with this right now. What I want you to take away right now is kind of a superficial chunk of information. I want you to understand that DNA is double helix. It is replicated in a semi-conservative model, and it is the carrier of genetic information. We're going to get deeper into DNA into the future, but for the time being, that's what I want you to know. The model that is actually used, and then the two models that were kind of dismissed as there wasn't really any evidence to support them. Now, as a quick visual for what that actually looked like, well, what Mieselson and Stahl did was they used radioactive nucleotides, which we've seen this before. Hershey and Chase used radioactive phosphate. or phosphorus and radioactive sulfur. Sulfur is going to be found in proteins, whereas phosphorus is only going to be found in DNA. Now, Michelson and Stahl used um, another radioactive atom and two different isotopes of it. They used radioactive nitrogen. And what they did was they said, okay, well, we're going to grow this DNA in 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 a nutrient bath or broth that has 14N, so one radioactive isotope of nitrogen, and 15N, a different radioactive isotope. And what we're expecting to see is that as these generations proceed, as we get more and more generations, they did a total of 14 generations. And what they saw was that despite starting with a basically no N14 and nothing but N15, they found that these successive generations incorporated this other isotope. So what they saw after 14 generations was a greater emphasis on this radioactive nitrogen, or this 14 labeled nitrogen. And that might seem like, wait a minute, that's a little bit odd. Well, let's think about it in terms of the only generation that had this isotope was the very first one. So 100% of the DNA had this 15N. Then in generation number two, well, that's where we're incorporating. So you could think about it as this dark red is 15N, and this pink or light red is 14N. So what percentage of our strands right here, what percentage of our strands there have 14, or sorry, what percentage of our strands are labeled with the 14N? And just to give you a little bit more direction, we have a total of 4 strands. What percent are 14N labeled? 50. Okay. Now let's imagine this right here. Let's take this strand where I'm going to, again, I'm going to do some erasing. This strand right here, where I cannot draw a star, so I'm just going to draw a smiley face. Okay. With that strand, what we're going to do is we're going to use both of those strands, both of those single strands as templates. Does everyone follow me there? So one template, one temp, will be 15N. One template will be 14N. All of the new strands will be nothing but 14N. So I'm going to label this as NS, new strand, 14N. Then NS, 14N. So now in our next generation, in this circle, I have again 4 total strands. Does everyone vibe with that? We've got 4 strands. What percentage of them have the 15N isotope of those 4 strands? 25%. So that's what we see right here. What's going to happen to this 25 percentage, or the percent of our strands that have that 15N? It's just going to basically get cut in half and in half and in half and in half every successive generation. Can I get a Y, or yeah, can I get a number if, okay, cool, I gotcha. And a letter if, okay. I noticed whenever I prompted that question the other day, somebody threw the number 0 at me. I was like, wait a minute. Is that a letter or is that a number? So the point of this is just to show that when we started with DNA, where 100% of it had this 15N, this specific isotope, then in every successive generation, we saw that the abundance of that isotope go down lower and lower and lower. So initially, 100% of our isotope was that 15N. Then in our second generation, 50% of the strands of DNA were 15N, then 25, then 12. If we went to generation number 14, well, then we're talking about, let's see, divided by two, we're talking about like 0.05% of the strands of DNA have that 15N isotope. The important part about this is what I want you to recall and understand is that isotope or those two isotopes were used to identify, well, this came from the parent and this is a newly synthesized strand of DNA. Okay. So again, anything in this class, if you've got questions about it, I'm more than happy to meet. I plan to have office hours tomorrow, so if you want to join that Zoom meeting, more than welcome to, or just send me an email and we can find another time. Okay, so this brings us to the central dogma of molecular biology. Sometimes you'll see this referred to as the central dogma of biochemistry, and that is DNA goes to RNA, which goes to a protein. This used to be something very linear and simple, that it was DNA goes to RNA goes to protein. DNA is transcribed in a process known as transcription. To RNA, and then RNA is translated to a protein. Now, what's a little bit to add a little bit more detail to some of these arrows, this first arrow that's kind of circular, this one represents replication, DNA replication. So DNA serves as the template to make more DNA. This this arrow that goes RNA to RNA, well, RNA has catalytic activity and can carry out specific chemical reactions itself. And then RNA can be converted from itself to DNA through a reverse transcription. So reverse transcription. So this initial idea of, oh, central dogma of molecular biology, it starts at this and ends at this. That's a nice idea, but it's far too simple. And that's all a conspiracy cooked up by biologists. Yep. To make things more difficult for college students. That's the plan. So if you keep this straight, what I'd like to do kind of is to reference back to the central dogma of molecular biology and biochemistry so that we can kind of put things into context. Now, with that in mind, let's talk about water and buffers. Now, we will not be making any buffers, but I will want you to know buffering range and basically managing what a buffer is and how it functions. I think that in Gen Chem 2, everyone probably made more buffers than they thought they ever would make, especially in that lab. But that's done. So we'll move forward. Now, water. Mammalian cells are 70% water. 2/3 of our water is inside our cells, and 1/3 of our water is outside of our cells. Now, why is water important? Well, it's a solvent for biological systems. An aqueous environment is primarily or predominantly water. Water enables hydrophilic compounds, which dissolve in water, to interact. Examples of hydrophilic compounds or water-loving compounds are polar compounds and ionic compounds. Now, I don't know about you, but I personally struggled in organic chemistry with classifying things as polar and nonpolar. I remember I felt like I was just guessing. Be like, hexane, ooh, polar. No, okay. Hexene, ooh, uh. I struggled with that, and I have no bones about that. Whenever I think of polarity, what I think is the most helpful thing for me to do is to look at and think about the periodic table and the Pauling scale of electronegativity. If I can see an atom like an oxygen or a nitrogen or a fluorine, well, all of those, I know that fluorine's my most electronegative element. Nitrogen and oxygen. Okay, well, those chances are going to have at least one lone pair of electrons. That helped me tremendously whenever it came to classifying polar compounds and or polar regions of a molecule. Um, and that's also helpful specifically those atoms because what we'll talk about a lot in this class and what's very important for biochemists is hydrogen bonding. So, polar compounds and ionic compounds love hydrophilic environments. They will interact with water till the cows come home. Hydrophobic compounds, by comparison, really don't like water. They are hydrophobic. They are scared of water. If they were driving a truck to their local high school, they would not be able to get the bumper sticker that says ain't skeered. They would have to get the bumper sticker that says skeered. Now, it seems like that's something that maybe was just popular when I was in high school, but there we go. So, hydrophobic compounds do not interact with water. These are your non-polar compounds. Now, a polar compound has an unequal sharing of electrons. So, with polar compounds, we're going to determine that and classify those based on electronegativity, the tendency to attract electrons within a bond. Which atom wants these electrons more? Well, is it the carbon? Is it the hydrogen? Is it the oxygen? It depends on that electronegativity. I'm not going to give you electronegativity values and ask, is this a polar bond? Instead, mostly what we're going to be doing is classifying a bond as being a hydrogen bond or not a hydrogen bond. And then if it's a polar bond, we'll be able to determine that based on something like the presence of any lone pairs of electrons or um, in the event that the molecule is asymmetrical. So electronegativity is the tendency to attract electrons. Just to give you a little bit of a refresher, electronegativity values range from, I think francium is like 0.7 and fluorine is 4 or 3.9999 or something like that. And this is all relative. This is the Pauling scale of electronegativity. So appropriately, well, who's the person who kind of cooked the idea up? Well, it was Linus Pauling in 1932. Linus Pauling, who was a very successful scientist, and now there's a research institute at Oregon State University that kind of continues his work into nutrition. Because in the later portion of his life, he really got into investigating like nutrients that people have, nutrient deficiencies or nutrients that people are consuming an adequate amount of. He also, I believe, coined the term mega dosing, which is used typically when it comes to like vitamin C. Um, right now, I'm mega dosing a Monster Energy Zero Sugar. Um, it's got a mega dose of, let's see, vitamin B12, 500% of my recommended daily amount. And just so you guys know, it's zero sugar, so you know it's healthy. Now, with that to say, Linus Pauling, Pauling scale of electronegativity, identified atoms that, oh, well, this one wants electrons more than another one. Now, like I said, I'm giving you these numbers just to kind of set your barometer. I don't expect you to memorize these because I think if most folks in this class, if I gave you a question, a blank question that was, what's the most electronegative element? I think most of you guys are going to be able to come up with fluorine. And then if I asked, like, what about some other ones? You'd probably come up with, well, I gave you parameters like something with lone pairs of electrons. You'd be like, ooh, oxygen. Oxygen loves to have two lone pairs of electrons. Um, so whenever I think of electronegativity for this class, my head goes straight to nitrogen, oxygen, and fluorine. And I go to those because those are important for hydrogen bonding. Okay, so dipole, a dipole. Polar bonds and molecules are also known as dipoles. Remember, polar means an unequal sharing of electrons, and electrons have a negative charge. The atom with the greater electronegativity attracts the electrons, giving that atom a partial negative charge. Yesterday, I asked about functional groups, and there are a couple of functional groups that people came up with, and one of them was an aldehyde. An aldehyde functional group. Okay, so the looking at this business end of an aldehyde, the atom with the greater electronegativity attracts the electrons, giving that atom a partial negative charge. I think almost everyone would agree, based on what we've just been saying about nitrogen, oxygen, and fluorine. Well, this oxygen's got lone pairs of electrons, so that's where our partial negative charge is going to come in. Now, this is just to kind of jog your memory a little bit. That partial negative charge, I'm not intending to ask this as a trick question, but is that a negative charge? And this was the sort of question I always got stumped on. No, it's not. You're right. I would be like, well, it's partial negative. Yes, but is it a negative charge? No, it's not. It's a partial charge. So whenever we talk about ions, a sodium ion doesn't have a partial positive charge. A sodium ion has a positive charge. So if you are confronted with that sort of like mental gymnastics whenever you're taking a test or a quiz or something like that, think about that. I hope that's helpful. Now, if there's something that has a partial negative charge, that means that this carbon over here is going to have a partial positive charge. Those partial negative and partial negative positive charges are going to be attracted to, well, opposite partial charges. And that's important for, well, how are these functional groups going to behave in in a solution? Each atom of the bond has a different partial charge, thus the term dipole is adopted. We visualize a dipole like that, where we've got the kind of dagger. I always like this, where I'd see, oh yeah, that is a positive. Oh, sweet. That makes sense to me. Positive right here. Boom. That's where my partial positive charge is. Okay. Methane is nonpolar because, well, it has nonpolar bonds. Specifically, the electronegativity of carbon is 2.5, hydrogen is 2.1. So if you're ever asked to do that, or you were historically asked to do that sort of calculation, you do 2.5 minus 0.1 or 2.1. Okay, our difference is 0.4. So it's not a big electronegativity difference. In addition to that, well, it's a symmetrical molecule. This is tetrahedral. So all of these bonds, or all of these adjacent atoms, are basically pulling in the exact same direction. I always kind of visualize this as like some sort of three-dimensional game of tug of war that's taking place, where everyone is pulling with the exact same strength and nobody's going anywhere. So our geometry and those individual bonds classify methane as a nonpolar molecule. Okay, now, yeah, carbon hydrogen bonds are never polar. Water, by comparison. Well, water's your classical dipole because that's usually the way that people introduce dipoles, is they say, here's a water molecule. We got partial positive charges and partial negative charges. Dipole, done. The electronegativity differences, 3.5 and 2.1. That's a significant difference. It's also an asymmetrical molecule. It's non-linear. We've got a bent structure here due to the fact that we've got lone pairs of electrons. Boom, boom. boom, boom. Those take up huge amounts of space, and they're going to force these rascals down. Okay, so those polar bonds are not going to cancel one another out. Instead, we're going to have something that, well, they're kind of constantly moving around. And there's our daggers to really nail home the idea that this is a polar molecule and has a dipole. Carbon dioxide, CO2, also nonpolar molecule, but because it's linear. And well, let's see, 3.5. Yeah. I mean, those individual bonds, they're each polar bonds, but they cancel one another out. And they're kind of right. Well, yeah, they're polar enough, I'll say. Okay. Hydrophilic compounds. Non-polar or non-ionic polar molecules. Oh, there we are. Compounds dissolve in water because dipole-dipole interactions. So non-ionic polar molecules will dissolve because they will interact with those dipoles. Ionic compounds readily dissolve in water because of ionic or ion-dipole interactions. So if we took something like sodium chloride, I always like this was really helpful for me to visualize, okay, there's a sodium ion that has positive charges, and just imagine that this is another sodium ion right here. Well, that's going to reorient a water molecule. So the partial negative charge is in close proximity to that. The partial positive charges are going to be positioned, well, so that they're close to that chlorine ion or that chloride ion. So basically, these sodium ions and chloride ions are almost picked off one by one, and they get hydrated by water molecules. They get surrounded. It's almost like if you got a bunch of ants and you threw a brownie on top of an ant pile. The ants are going to come up and get a little piece, and they're going to go back. And that's kind of my visual, the ants being the water molecules and the brownie being the NaCl chunk. Okay, hydrophobic interactions. Non-polar substances, they don't like water. The hydrogen bond network of water reorganizes to accommodate the non-polar solute. Okay, so what we're going to have is this right here that I'm putting a circle around. This might, let's see, I'm just making sure that I don't actually have it on here. There's a little bit of a clue as to what this is, but could anyone tell me that molecule that's in the middle? What what's a good name for it? And you don't have to have the specific name, but a class. Phospholipid, super close. Yes, it's a lipid. And do you guys, I was I don't want this to sound like any sort of disrespect or anything like that, but if somebody had said it's a carbony, Carbony, which is not a molecule, if somebody had said that, I'd been like, that's pretty close. But you guys are like right there. If you were golfing, you landed the ball on the green with lipid and phospholipid. This is a lipid. Specifically, this is a fatty acid because this it's got an alkyl group, this hydrophobic alkyl group, and then it's got this hydrophilic head group. Miracle's responsive phospholipid, you are like right on the cusp of it because a phospholipid includes one of these because a phospholipid has that hydrophilic head group. But there's a little bit more to a phospholipid than that. But I love you guys' responses. Thank you very much. Good job. Um, Essentially what's going to happen is if you were to take a single fatty acid and throw it in water, this is effectively what's going to happen. This hydrogen bond network that exists within water is going to basically go from this state right here, where, hey, all these water molecules are kind of together. They are going to reorganize and, well, they're just going to try to keep like a distance between themselves and the hydrophobic alkyl group. The head group, oh, they're fine with that. They like that because that has, oh, look at that, a negative charge. So what do you think is going to happen is, well, these positive charges here and here, they'll be in close proximity to that because they're cool with that. Now, that's if you have one of those molecules. If you got more, well, then what's going to happen is those individual fatty acids are going to cluster together. Those are going to cluster together, and these water molecules are going to kind of surround them. And they're going to say, okay, well, that's cool. You guys get together, and we're just going to kind of we're going to watch you because we don't really trust you. We don't know what's going on, but I don't know, we're all polar, you're non-polar. What's going on there? So a non-polar solute organizes that water. So it causes that water to become a little bit more ordered, which is an increase in order. That increase in order is, and you might have been thinking like, we said enthalpy, entropy, and free energy yesterday. Certainly, we're not going to talk about those things again. Please, please. Well, I'm sorry, I'm bringing it back up. Yes, that increase in order is a decrease in entropy because those water molecules have become the way that I think is helpful to think about it is when you have three kind of awkward clusters of water molecules around these, going from three of them down to one of them with, you got these clustered water molecules around them. That is more organized because it's kind of like whenever I'm, organizing something. I've got a bucket of crayons. That's all I can think of right now, I guess. And I'm going to say, okay, well, I've got red crayons over here. I've got green crayons over here, blue crayons over here. I'm going to put them all into my one bucket. It's more organized because I don't have like a pile over here, there. Um, but that's a decrease in entropy that we're observing there. As a result, the nonpolar substances tend to aggregate to reduce the decrease in entropy. Now, this is ultimately known as the hydrophobic effect. So we have gone from talking about polarity of compounds and polarity of molecules to now getting into, well, what are the implications of that in an aqueous environment? What does water do in an aqueous environment? The hydrophobic effect is where hydrophobic or nonpolar things cluster together to kind of form like a safe space for them. Those are exhibited as micelles as well as lipid bilayers. Now, a lipid bilayer, the central part of it, super nonpolar. On the top side of it, or the bottom side of that lipid bilayer, that's going to be an aqueous or polar environment. Now, we don't live in a world of simply things that are polar and simply things that are nonpolar. Instead, we've got things that are combination. A good example of that is a fatty acid. It's got a hydrocarbon tail and a polar head group. There are molecules, lots of molecules, that are known as amphiphilic molecules. That, well, they are they have polar regions and nonpolar regions. For instance, the amino acid phenylalanine right here. Well, it's got a nonpolar portion, but then it's got a positively charged and a negatively charged group. So it's got a polar or it's got two polar regions and a nonpolar region. Another one known as phosphatidylcholine. Well, it's got a nonpolar group and then it's got polar regions, sure enough. Now, there's a point whenever it comes to kind of classifying these and, well, how are they going to interact? Well, they're capable of interacting with polar and nonpolar partners, but it's important to think about this molecule right here, phosphatidylcholine. Phosphatidylcholine is a phospholipid. It's known as a glycerophospholipid. I'm going to put A and B of this molecule. I've got two regions. I've decided to separate it simply as two different regions, region A and region B. And before you answer, I will let you know that phosphatidylcholine absolutely loves to be inserted or embedded within a lipid bilayer. Okay. And what that looks like, to give you a little sense of it, it's going to look, it's going to be kind of dramatic, my drawing here, but it's going to look a little bit like this. Okay. Now, my drawing is not very good. It's great. My question for you guys is when you look at my rendering of that molecule being embedded in a lipid bilayer, this portion, which I'm putting an X by, that is, well, that's definitely inserted and in a very hydrophobic region. Where I'm putting a Y, that is in, or that is exposed to a very hydrophilic region. Now, my question for you guys is region A embedded within the membrane, or is it region B, which is embedded within the membrane? And if you're unclear on the terminology that I selected, I'll come back to it. For the moment, I need to drink some zero sugar L-carnitine and taurine. Okay, I'm seeing A responses. Great job. Essentially, what's going to happen is this portion of the molecule that will be, I'm going to erase the X and I'm going to put an A here. That will be embedded, inserted, and kind of anchored into this membrane. Now, that's important because when a molecule like phosphatidylcholine gets embedded or anchored into a membrane, well, then that's a way for cells to communicate. It's a way for a cell to signal, hey, I'm under distress. There's a pathogen present. So now I'm going to display a bunch of phosphatidylcholine. Or it's just a way for cells to communicate and say, I'm this type of cell. You're that type of cell? Great. And then also, like, I'm this type of cell. I've got this glycerophospholipid, and I am waiting for the right protein to come along and grab onto that. That's something that we're going to get more into later on this semester when we talk about lipids and cellular communication. But for the time being, what I want you to know is there are polar molecules that have a role in a cell, there are non-polar molecules, and there are amphiphilic molecules that have a role in the cell. Okay. Let's see. It's 2:53. So close to halfway through. That's good. That's good. Um, okay. Non-covalent bonds or forces. Non-covalent bonds determine the complexity, or actually, let me take a pause. I'm going to jump back here. Um, if you would like to answer this question as a private message, by all means. I would like to know on a scale of 1 to 3, 1 being, hey, I've got all this. I'm happy for the refresher, but I've got all this. And 3 being, I'm I feel like I'm falling behind. I feel like I didn't have a strong grasp of this material previously. Um, there we go. Just respond, and you can send it as a private message. Just kind of want to get a sense of where people are. Okay. 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 Um, Let me just tell you that I'm really happy whenever I ask that sort of question. And my thought process is there's basically 3 answers, 1, 2, or 3. And the very first response I get is not a whole number. I always like that because that is, to me it's this is maybe like silly, but I look at it and I'm like, Joel, you do not see the world the same way as everyone. Not everyone sees the world the same way as you. So there you go. And I like that interpretation because there is absolutely gray area for stuff. Um, okay, so I'm seeing lots of 2s, and that's absolutely fine. Um, what? So I'm hoping that as we move through this, um, tomorrow and Friday, that you'll kind of get your bearings about yourself and you'll feel a little bit more confident in the material. Lots of this is, I mean, in a sense, this is kind of a crash course of Gen Chem 2. It's like a peppering of, hey, what were the important concepts of Gen Chem 2 and important slash tricky concepts? Let's grab those and just fly ahead. Um, Tomorrow we are going to get into amino acids. And so I think that will help because it's like taking knowledge and applying it. So I'm hopeful that that will give you a little bit more clarity. And then a lot of this is just kind of new terminology. Okay. So thank you guys very much for those responses. Okay. Non-covalent bonds determine the complexity of molecular interactions within and between biomolecules. Non-covalent bonds as opposed to covalent bonds. So just to revisit that, a covalent bond is where you have sharing of electrons, pretty much an equal sharing of electrons. Whereas a non-covalent bond is where you have an unequal sharing. Um, I think that when you think about non-covalent interactions, I always like to pick something like a hydrogen bond as my model for a non-covalent interaction. Alternatively, does anyone remember London dispersion forces? Super weak. Or induced dipole? Super weak. I like to think about an induced dipole, basically CH4. Okay, we've got. I always like to think of CH4 or two CH4 molecules and how they interact with one another. So, just as a little bit of a reminder, I'm drawing an arrow to a covalent bond. Okay. Yeah, got it. Carbon to hydrogen. That's a pretty boring bond. Those electrons are pretty evenly shared. If you think about electronegativity, neither one of those atoms is super electronegative. But then if we say, all right, not necessarily within a molecule, but how does molecule A here and molecule B here, how do those two interact? Well, they're going to interact with an induced dipole. What do I know about these two molecules? They're both nonpolar molecules. So those two nonpolar molecules interacting through an induced dipole, super weak interaction. Okay. So I always like to think about this and this versus that. So I'll write strong, weak. It's easier to separate two methane molecules from one another than it is to separate a carbon from a hydrogen. Okay. So they are 10 to 100 times weaker than covalent bonds because your non-covalent interactions are on a spectrum. Some are stronger than others. Those those induced dipole interactions, super weak. Single interactions are typically not sufficient to hold two species together. The 3D structure of most macromolecules are determined as a result of the collective influence of many weak individual interactions. So that right there, that sentence or that bullet point, that's a mouthful. The 3D structure. What I want you to take away from this is that when we think about biological macromolecules, and I'm just going to give you an example, a protein. Actually, I'll give you a specific example. Is anyone familiar with, I was about to say familiar with hemoglobin? Hemoglobin is an oxygen transporting protein, essential for me to do this. Ooh, it's very helpful. I really like hemoglobin. Now, has anyone ever heard of sickle cell anemia? Sickle cell anemia is a disease state caused by a mutation in hemoglobin. That mutation causes, so I'm going to write this as, well, yeah, sickle cell that mutant form of hemoglobin that causes sickle cell anemia results from the protein taking on this weird or this different shape. So the 3D structure, that what something looks like in 3 dimensions, is super important. Now, you touch on this in Gen Chem 1 when you talk about VSEPR theory and you look at linear versus nonlinear molecules. But there you're talking about, 5 atoms, 6 atoms, something, you're talking about something small. When we're talking about a protein, we're talking about hundreds of amino acids and hundreds of atoms making up that large molecule. So that's the point here. And so that's the magnitude of what we're talking about. And it's collective influence of the many weak interactions. So when it comes to the structure of a protein, we, strange as it may sound, we care about this. We care about these induced dipoles, these overall weak interactions, because they contribute to the 3D structure of a protein. Okay, non-covalent bonds are reversible. Non-covalent interactions are very specific. The size, shape, and type of interaction all must be correct for binding to take place. Now, within biomolecules, yeah, we've got those covalent interactions. These are numbers I don't want you to commit to memory. I don't want you to think like, oh, I got to remember that a ionic interaction is approximately 86 kilojoules per mole. I don't want you to stress about that. What I want you to know is I want you to know that covalent interactions are as much as 20 times stronger, as much as 100 times stronger. What we will talk about maybe the most in this class are ionic interactions, hydrogen bonds, and dipole-dipole interactions, because those are the interactions that drive lots of protein folding. Now, to dig deeper into hydrogen bonding. Hydrogen bonding is one of the most important interactions in biological molecules. Hydrogen bonding is an interaction between a covalently bonded hydrogen atom in a donor group. So we've got the donor and a pair of non-covalent interactions on an acceptor. So I'll use this terminology of hydrogen donor, hydrogen acceptor. Uh, and it's important for you to remember, and we'll look at a couple of models of this in just a little bit, but important for you to be able to apply that terminology. So the ability of a group to act as a hydrogen bond donor depends on its electronegativity. The bond must be polar in a hydrogen bond and must have a free pair of electrons or a formal negative charge. One thing that I think is easy for most folks is to remember that carbon-hydrogen bonds, or actually, let me rephrase that. One thing that I've noticed over the years that people have a tendency to fall into a little trap on is they say, oh, as long as I see hydrogen, I'm talking about a hydrogen bond. Is that correct? Give me a number if that is correct or a letter if that is incorrect. That all you need? Great, great, great. If you have a hydrogen, that does not mean that you've got a hydrogen bond. More times than not, when you see a hydrogen, it's bound to a carbon, and that's going to be a covalent bond. Now, let me go ahead to this one. Common hydrogen bonds. This is a good visual of what they are. Your donor, what does your hydrogen bond donor have? Well, that's got the covalently bound hydrogen right here. What is that bound to? Well, it's bound to an oxygen or a nitrogen. It could also be bound to a fluorine, but the donor has the hydrogen. I think that's an important thing to remember. Or, well, no, I don't think that is. I know that's an important thing to remember. The hydrogen bond acceptor has oxygen, nitrogen, or also fluorine. So it will be important for you to be able to recognize which species is the acceptor and which species is the donor, the hydrogen bond donor or the hydrogen bond acceptor. Now, hydrogen bonds, even though we've got this number right here, a hydrogen bond is approximately 20 kilojoules per mole. Not all hydrogen bonds have that exact same strength. The reason for that is, look at this right here. Non-linear hydrogen bonds are weaker. The strongest hydrogen bonds have the donor atom and the acceptor 180 degrees away from one another. Another way to phrase that is these are directly across from one another. Okay, so think about this as, well, well, that's a straight line, but not really the angle that I was going for. So I'm going to try it this way. Nope. You get the point. Hydrogen, oxygen, straight line between them. They're as close to one another as possible. Hydrogen and oxygen, oh, yeah, it's a straight line, but it's at an angle. They're not directly across from one another, which weakens, or it makes that a relatively weak hydrogen bond. Now, to visualize hydrogen bonds that we'll talk about, we've got alcohol and water. Here's an alcohol, here's water. Here is a carbonyl and water. Here is a peptide and another peptide. DNA. DNA. Hydrogen bonds, strengthen those, stabilize those. Whenever I give you this sort of, or if I were to give you this structure and I were to say, okay, A, oh, not right, A or B, which one is the donor? Which one is the acceptor? The hydrogen bond acceptor. Okay, so let's jump back. Our hydrogen bond donor has the covalently bound hydrogen. So our hydrogen bond donor, which is the donor? Well, B is the donor. Therefore, A is the acceptor. Okay. Now, hydrogen bonds, well, they're going to strengthen proteins. They're also going to stabilize, or they're going to stabilize and strengthen different structures of proteins as well as DNA. So that's kind of your crash course on hydrogen bonding. Let's see, think about that as you need to know definitions now. We will be applying them as we proceed through the semester. We'll be looking at specific applications. If you can identify which species has the hydrogen and which has the electrons, that's going to be very helpful. Okay, now we're moving on, and I believe that this is, let's see, let me zoom out. Oh, no, that's zooming in. Where we are now, nope, that's not what I wanted to do. That's what I wanted to do. There we go. What I'm going to do here is I'm going to give you a little bit of a rundown of this, and then I'm going to work through some examples of what is important to take away from this. So, weak acids and weak bases. The difference between weak acids and strong acids is that weak acids only partially dissociate. I remember for the longest time struggling with that. I was like, oh man, how can I remember what are possibly the weak acids? Well, as soon as you remember the 7 strong acids, then you know every single other thing is a weak acid. Whenever I learned that, I was like, oh, I don't know if anyone else knows this. Man, whew, I've just got a treasure trove of knowledge. And I feel like if I share that, that's like me cheating. And so I kept that to myself because I didn't want to cheat. So, but I've since learned that no, that's not cheating. That's just the information that everyone else already probably had. So, strong acids, weak acids. In biology and in biochemistry, we're dealing with weak acids and weak bases. We don't really talk about strong acids very much. Um, and the reason for that is just because they're not, because most everything in a biological system is buffered. So it resists any sort of major change in pH. So partial dissociation, HA goes to H plus A, which really, it actually kind of goes to plus HA because that stays intact. We have an equilibrium constant, erase this, where we have our products over our reactants. And for that, we have a Ka, it's the ionization constant for a weak acid, HA in water. Now, typically what we are going to talk about in this class is we're going to talk about pKa's. I'm going to jump over Ka's, and I'm just going to focus on pKa's. What I want you to know, what's important for you to know, is that the smaller the pKa, well, the more acidic or stronger the weak acid is. The larger the Ka, the more acidic or stronger the weak acid is as well. The proton has a stronger tendency to dissociate. So if you have a very large pKa, a large pKa means most of the acid, most of weak acid will remain HA, remain intact. Whereas if it's a very small pKa, well, then it's more likely to dissociate to a greater degree. In other words, the dissociation is pushed to the right or to the hydrogen ion. Some examples of pKa's. Let's take phosphoric acid, H3PO4. And that's really going to be kind of the example that I build on. H3PO4. That has one form, a second pKa, a third pKa, and then it's actually got a fourth pKa because it has 3 ionizable protons. So H3PO4, what's another term for that type of acid? An acid with 3 ionizable protons. Add that to the chat. Or if you have your video on, you can just go, yep. Hydronium, close. What I was looking for is triprotic acid, compared to, say, a diprotic or a monoprotic acid. On the list here, can anyone add to the chat a diprotic acid? So something with 2 ionizable protons. Close with the H. I was looking for the H2CO3. So H2PO4 minus, that does have 2 protons that it can still lose. But let me rephrase. I needed to correct my kind of question. When something's fully protonated and it only has 2 protons, find one. So that was my bad on the question there. Okay. Now, if we have a triprotic acid, okay, I'm going to zoom through the next few slides and then I'm going to come back to this one. Zoom through. Relationship between pH and pKa, we talked about that yesterday. The Henderson-Hasselbalch equation, we talked about that just a tad yesterday. We're going to talk about it a little bit more today. Then a titration curve. The last bit of this is all titration curves. So we are going to work through drawing a titration curve for phosphoric acid. Okay, that's what I want to spend the next little bit of time doing. But before we do that, what I want to do is I want to introduce you to all of the different forms of this H3PO4. So one form, it's fully protonated. It is H3PO4. Now, to give you a visual of what that looks like, phosphorus, oxygen, 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 H, H, H. H3PO4. I'm sorry, I had to pause because I can hear my kids and my wife, and I'm trying to figure out if they let the dog out. And the dogs drive me crazy because when they get out, they sprint and they just go. Maybe they're telling me something. Okay. So here is our fully protonated form of H3PO4. Now, my question for you guys, and this is not a trick question, but this is an important question for you to kind of recognize because this is going to be valuable as we proceed. What is the charge of this molecule that I'm putting a big circle around? And I like asking this question because I can tell everyone's thinking about this. Drew, you're absolutely right. Zero. It has no charge. I bring this up because this is two things that are true and they are not contradictory of one another. This molecule is fully protonated. It has a neutral charge. Those are both true statements. What happens from time to time is that people will think, oh, well, if something's protonated, then it must have a positive charge. And that is not necessarily the case. So this is an example of a molecule that is fully protonated. Now, this H3PO4, are there any other protons that it can take on? And still kind of exist? No. There's no other place to put another proton there. So H3PO4, fully protonated, 3 protons, 0 charge. Now, if that molecule loses a proton, H plus, now it is H2PO4 minus. If it loses another proton, H plus, now it's HPO4 2 minus. Last but not least is where we get to the phosphate ion. It loses its last proton, and that is PO4 3 minus. So we have 4 versions of this molecule and 3 protons that it can gain or lose. Now I'm going to take these pKa's and I'm going to put them next to these, basically these dissociations. 2.15. The next one is 6.82. And then the last one is 12.38. And you know, a moment ago, I said that it should have a 4th pKa, and that is incorrect. I apologize. It has 4 forms, but 3 pKa's. So I misspoke a moment ago, and I apologize for that. Now, with these pKa's, yesterday we were talking about pKa's, and I asked the question, well, if you took this molecule and put it in a solution of this, does the pKa change? And the consensus on that was no, it doesn't. Because the pKa is a value that is, and I love using this word because it makes me feel really fancy and smart, intrinsic. It's an intrinsic value that's a measure of how much does this molecule want this proton. So pKa's don't change very often. Under different conditions, yeah, they might. If the molecule changes, the pKa is going to change. But here, what we have is I'm going to ask the question, what if I made a solution? And the solution's pH was, let's go with 9.47. I could have gone with 9. I could have gone with 9.00, but I didn't. I wanted to keep you guys on your toes. 9.47. Could anyone tell me which different form of this molecule? And you know what? I'm going to make it a multiple choice question. A, B, C, D. I'm looking for which of those 4 different forms of this molecule is the predominant, or maybe the most commonly found version of that molecule in that solution. Drew's voting for C. And got to be honest with you, Drew's right. Drew's right because the pH of my solution, it's right here. It's between my 2 pKa values. And so the predominant form is that HPO42 minus. Now, if I erase all this, I'm going to go to draw a titration curve here. And hopefully that will give you a little bit of a visual. And you know what I'm going to do? I'm going to take a second. I'm going to add a blank slide because then I will have a slide without any sort of uh, markings on it so I can, play around with more space and have something that, there we go. There we go. So got a titration curve. It's a y-axis. There's my x-axis. My different forms of my molecule. H3PO4. Then I've got H2PO4 minus. Then I've got HPO4 2 minus. And then last but not least, I've got PO4 3 minus. So those are my 4 different forms. I like to, anytime I'm drawing a molecule, I like to know what are my possible molecules. I'm going to go back 2.15, 6.82. 12.83. Okay. So those are my 4 different forms of the molecule. I like to start out and ask myself the question, which of these is going to be the form of my molecule? A, B, C, or D if I have an extremely acidic solution. And so I always like to think about that as, okay, in an extremely acidic solution, what do I know that I have a bunch of? And I'm talking like pH of 1 or pH of 0.5. If I have a solution with a pH of 0.5, I've got basically an infinite number of these hydrogen ions. So everywhere that a hydrogen ion can possibly go, it's going to. Now, of these 4 different forms of my molecule, which has already kind of accepted all of the hydrogen ions that I can possibly accept? Is it A, B, C, or D? A. You're absolutely right. my fully protonated form is A. That is the form that's going to exist at a pH of, well, 0.5. So what I'm going to do is on my y-axis is I'm going to label this as pH. And that's going to range from 0 to 14. I'm just going to make it easy on myself and label it that way. Now my x-axis is going to be hydroxide ion equivalents. These hydroxide ion concentration equivalents are going to be basically whenever I've taken care of one group. So what I'm going to do on this is I'm going to do a couple of different things. The first thing that I'm going to do is I'm going to put 3 dots in this plot. Here's one. Here's another. And then last but not least, here's another. Could anyone take a guess as to what those dots represent or correspond to? Those dots, well, midpoints, also my pKa's. So I've got 3 different pKa's, 2.15, 6.82, and 12.83. If you remember drawing a titration curve, I'm going to switch colors just so I can make it more fun. I can go to green. There we go. There we go. And some regions of that titration curve are better than others. But this gives me a rough titration curve for this molecule. I've got my 3 points, my 3 pKa values, and I've got 4 different forms of it. Now, if I have a pH, if I were to, so the way that I always like to think about this is I make a solution with a pH of 0. And then what am I going to do? I'm going to add, let's go with KOH, potassium hydroxide, a strong base. I'm adding base. I'm adding more and more base. And what's going to happen is for every base molecule that I add, one of these H3PO4s is going to be converted to H2PO4 minus. I'm going to have an equivalence point where basically 50% or 0.5 of my molecule is A, and 0.5 or 50% of it is B. So my question for you guys is, if 50% of my molecule is A and 50% is B, what is my pH? So before you answer, let's say that I have 1,000 copies of H3PO4 in this solution with a pH of 0. So here's my solution. pH equals 0. 1,000 of them look like H3PO4. And what am I going to do? I'm going to add KOH to this. Whenever I add base to that solution, what's going to happen to my pH? It's going to creep up. My pH is going to go up. Maybe not dramatically, but it's going to slowly but surely go up. Let's say that I've added enough base where 500 molecules, 500 of the 1,000 are H3PO4. And 500 are H2PO4 minus. Does everyone follow that? Does everyone visualize that point? Basically, I've deprotonated half of the molecules. Not completely deprotonated, but I've removed one proton from half of the molecules in my solution. My question for you guys is, my pH is no longer 0 because I've added base. So what is my pH where half of them are H2PO4 minus and half of them are H3PO4? I'll give you a little spoiler. The answer is on the screen. It's 2.15. So this 0.5 is where I have half of them that look like A and half of them that look like B. Okay. So now, and I'm going to ask you like 2 more questions, then we'll call it a day, because we're going to pick this stuff up tomorrow. Let's say that now we have let me phrase it in a different way. This is basically the same. I'm about to ask the same question, but from a different angle. The pH of my solution is now 12.83. And you can use the letters down here. These A, B, C, and D. When the solution's pH is 12.83, what A, B, C, and D, what molecules exist in my solution? Again, I started with 1,000, now I got 500. I got C and D, I got D. The correct answer is I've got C and D. Because my pH of my solution over here is equal to this pKa. Now, again, at this point here, I'm sticking with this idea that I've got 1,000 of these molecules. I'm going to say 500 are PO4 minus, or 3 minus, and 500 are HPO4 2 minus. Okay. How many of them? Keep in mind, I started with 1,000. How many of them look like A when my pH is 12.83? How many of them look like A? How many of them look like B? This is a question, and I see the chat, and you guys are right. Keep in mind, we started with 1,000 copies of this molecule. 500 of them are PO43 minus. Let's do the math here. We've used 500, or 500 of them are PO43 minus. That leaves 500. And then another 500 are HPO4 2 minus. 500 minus 500 is 0. So effectively, you're right. There's almost none. Effectively, the answer is 0. Effectively, none, when our pH is 12.83, basically none of it looks like this, and none of it looks like this. because it's evenly distributed between HPO4 2 minus and PO4 3 minus. Now, if we were to, and this is my last question for the day. I'm going to imagine that I started with, we go back to the beginning. I had 1,000 copies of H3PO4, and I added into this solution with a pH of 0. I slowly but surely added potassium hydroxide. My pH very gradually went up. The H3PO4 got converted to H2PO4 minus, which then got converted to HPO4 2 minus, which then got converted to PO4 3 minus. Let's say that my pH, the pH of my solution, is not 12.95. I'm going to give you 4 different options. Okay, let me see how to write this out. HPO4. No, 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 no. I'll phrase it this way. Which is the most common form of my molecule when my pH is 12.95? I'm getting lots of responses of D. You're absolutely correct. Okay. Which is the second most common? C. Third most common? B. Pretty reasonable. Fourth most common? A. A and B, the concentration of those two is infinitely low. But, as is the case with any sort of scientific endeavor, you have to say, well, it's not absolutely zero. More than likely it's very close to zero, but it's not absolutely zero because there is a instantaneous or a moment of an instant where that H3PO4 can exist. Okay. So what we worked through here was basically a kind of a roundabout way of talking about the Henderson-Hasselbalch equation. What I did not do here was integrate or utilize like log calculations. And what is the precise concentration of this? We'll get into that a little bit on Friday. I've got the PowerPoint that's for tomorrow up, and we're going to look at a couple of amino acids. We're going to start with our classification of amino acids and look at how they, well, one, how they can be classified. And then we're also going to talk about how they change because all of this right here is important from a perspective of, oh, well, what does phosphoric acid look like? But we're using this all as a vehicle to understand what amino acids look like at different pHs. So with that, I'm done. I want to tell you that I hope you have a great afternoon. Thank you guys for coming to class. Thanks for engaging. I hope that I don't lose my voice. Usually the first week of classes, whether it's summer or long semester, end of the week I lose my voice, but then I get it back. So I hope that you haven't grown tired of hearing me talk quite yet, because we got like 20 more days of it. At any rate, I will post this recording a little bit later today. I hope that I'll have it before 10 p.m. I think last night it was by like 10:30. But anyway, thank you guys very much for coming. I hope you have a great afternoon, and I will see you tomorrow. Deals? 1, 2, 3, break.
I have a question.
Absolutely.
So,
Here, I'm going to stop the recording.
okay.